All right. Well, thank you, Aaron, and thank you everybody for joining this morning. I hope you can hear me okay. Um, Not make sure that I'm not muted. So I'm a new assistant professor. I just started about maybe a month ago in the biological sciences department, and coming from Houston at Baylor College of Medicine. So a lot of this work actually had been done prior to me joining. So some of you may be familiar with it if you heard my seminar. And by which at which point you can decide to leave. But if you decide to join, I may have a few new discussion points about my studies. So I'm going to give you a brief sort of background on what kind of led to this study on studying defects in the jaw, and we'll go from there. And bear with me with sort of this lengthy introduction. So as a postdoc, I've been working on mechanisms of osteogenesis imperfecta. This is also very well known as brittle bone disease. So it's a genetic disorder which is most prominently characterized by bone loss, severe deformities, and fractures. And it's a rare disease that affects approximately 1 in 15,000 individuals worldwide. And obviously, as you can see from this cartoon image to the right, it really does have a significant impact on the quality of life of these patients, whether if you're a mild to moderate patient who experience a lot of fractures, although they do end up growing up relatively normally, but in these severe to extreme cases, obviously you can see why they can have a pretty hard time during the course of their lives. And so one of the most well-studied mutations that lead to OY phenotypes is the dominant mutations in the genes that encode type 1 collagen, namely COL1A1 and A2. But since 2006, my previous lab and others have demonstrated a plethora of other gene mutations which lead to a similar spectrum of disease. And one of the genes that I'll tell you about is FKBP10, which is important for post-translational modification and cross-linking of collagen. And I just highlighted these different gene mutation clusters which affect various aspects of collagen biogenesis. And more recently, we found that there are mutations that affect osteoblast differentiation, the primary component of bone making cell populations, and also mineralization. And so these patients, as you can see, are present with skeletal deformities and fractures, which is one of the reasons why the focus of these studies have largely been prioritized for treating skeletal dysplasias. But as you can imagine, mutations in collagen and those that affect collagen biology can lead to a lot of different spectrum of phenotypic and pathogenic features. And one of the main ones that I'll focus on today is defects in the joint. So long story short, we generated a mouse model in which FKBP10 is specifically deleted in tendons and ligaments, so the soft connective tissues. And in these mouse, you can see that they're largely normal at birth, but they develop these contractures in their hind feet. And by X-ray, you can see how these look differently compared to their litter-mate controls. And this happens around approximately 2 months of age. And when we look at a more high-resolution micro-CT image, which is basically a 3D reconstruction of multiple X-ray slices, you can see that in the mutant situation, they have severely abnormal ankle joints and also ectopic ossification, so abnormal ossification or bone formation in the soft connective tissues of these mice. And so what happens histologically is that there is, in the case of the wild type situation, you have these normal tendon or ligament-like cell morphology, and these overtly differentiate into chondrogenic lesions, which further then is exacerbated into new bone formation in these sites, as you can see by Gaussian blue and hematoxylin orange G staining, as you can see here. And this is just to illustrate to the right that post-contracture, so after the phenotypical abnormality, superficially, you can see even further increase in abnormal chondrogenesis in these joints. And so what does this do to locomotor function? You can see that by open field assay, there is significant decreases in horizontal, vertical, activity as well as the total distance traveled, whereas the overall movement time remains relatively similar. And just to kind of breeze through this really quickly, and we can go back to it if you're interested, we performed gait analysis experiments where you can see the paw prints of wild type and mutant situations. And what you'll notice is that in the mutants, these mice tend to walk on their toes essentially, and they're unable to basically make contact with their entire feet during locomotion. And this is quantified here with showing significant differences, particularly in the hind feet as compared to the forefoot. So what is FKBP10 actually doing? We do know that during collagen biosynthesis, when pro-collagen molecules are actively translated into the ER, they undergo multiple post-translational modification events, including prolol-3 hydroxylation, which affects the helical domain of pro-collagen 1. And there is the telopeptide lysyl hydroxylation, which is at the ends of pro-collagen molecules prior to proteolytic cleavage. And this is regulated in part through a complex known as the lysyl hydroxylase complex, which is the enzymatic activity is contained by the enzyme LH2, and this is chaperoned by FKBP10. And so what happens is once these lysine residues undergo hydroxylation, lysyl oxidases recognize this modification and undergo covalent cross-linking modifications, which is essential for pro-collagen fibril formation. And essentially, for instance, in the case of tendons, this leads to tendon maturation. So just to give you a brief background on tendon tissue, we know that it's a tissue that transmits by mechanical forces generated from muscles to bone, and ligaments similarly does so connect bones to other bone tissues, and they have different functions, including weight bearing as well as stabilizing the tissue, and they're essential for locomotor function. And so, not to get into too much detail, but this cartoon diagram basically shows the orderly structure and the parallel orientation of tendon tissues, and they actually share quite a bit of similarity with muscle tissue, if you recognize that. And what this does essentially is this kind of hierarchy provides high biomechanical strength. And at the molecular level, we know that these tissues in particular are marked by the expression of a basic helix loop helix transcription factor known as scleraxis, which in its absence, we have poor tendon formation and tenocyte recruitment. And so this is, apologies for the long introduction, but this kind of provides the background for how I got into being interested in the jaw. And so this is just a very basic cartoon image of the human jaw, and you can see basically the upper jaw connecting to the lower jaw, and there is this disc structure that cushions the upper and lower jaw structures to minimize any kind of direct contact with the upper and lower jaw structures. But what we don't really think about very often is the tendons that connect the muscles, essentially the pterygoid and the masseter muscles, which are on the lateral side, to the condyle structure. And this is actually an image of the scleraxis GFP reporter mice in the TMJ, or the temporomandibular joint, which, as I alluded to in the previous slide, marks tendon cells. And you can see how the pterygoid muscles to the right insert into the bone through these scleraxis GFP positive tendon cells. And so this is a micro CT image where we segmented out the mandibular jaw from wild type mouse at 18 months of age, and you can see how there are similarities between the human and the mouse jaw. In the FKBP10 mutants, which I showed you earlier, you can see these extremely large temporomandibular condyle compared to the wild type situation. And so we became very interested in this, partly because temporomandibular joint disorders is a severe problem, is a severe clinical problem. It affects approximately 10 million individuals in the US with a higher prevalence in women than men. And it commonly features severe orofacial pain as well as limited mandibular mobility, i.e., it affects your jaw, where it affects you on a daily basis because you're using your jaw to be able to speak and eat, and these are two things that we cannot live without. And so, what we do know is that these diseases, or the spectrum of diseases, are associated with abnormalities of the tendons as well as the muscle or disc, and it really is not known what are the mechanisms that lead to temporomandibular joint disorders, and we need a better understanding to identify these mechanisms to be able to design targeted therapies. And so this is basically a whole mount image of skeletal staining. So all of the red here you're seeing is bone, and what you can see in the mutant situation is this abnormal bone formation in regions where the tendons directly attach to bone, known as the tendon to bone interface, or enthesis, if you like scientific jargon. When we quantify micro-CT, these regions by micro-CT at multiple time points, we see that at 1.5 months of age there's little difference, but by 6 and 18 months of age we see significant differences in the length of the mandibular condyle in the FKBP10 mutant situation. We wanted to examine whether this is specific to the mandibular condyle or has a general effect on overall mandibular geometry. And the short story is that we found no differences in the overall mandibular geometry, indicating that this is a phenotype that is relatively specific to the TMJ. And so, just to give you an idea of the histology, this is wild type condyle, and you can see the nice temporomandibular condyle and below. The disc and the upper fossa in the wild type situation. These structures are essentially absent in the mutant situation, and you can see degeneration of the disc, fibrillation of the articulating chondrocytes in the mutant situation, and overall massive abnormal bone growth in the mutant situation. And if we look earlier, and I'll try to finish this a little as soon as I can, but if you look earlier in the wild type situation, you can see various layers connecting the temporomandibular condyle bone to the pterygoid muscle, and this includes the tendon. And what you can see is that there is an expansion of the tendon to bone interface in the mutant situation, and this is quantified here to the right, and also marked by a population of alpha SMA positive cell populations that reside in the interface. And what we believe, and in data that I don't really have time to get into, we believe that there is basically an overt differentiation of these population of cells into the chondrogenic lineage or osteoblast lineage, leading to abnormal bone formation. So just to summarize, I've shown you that loss of function mutations in FKBP10 can cause skeletal and non-skeletal abnormalities in patients, and that tendon-specific ablation of FKBP10 can induce localized joint deformities in our mouse model, and that it also, in more recent data, regulates mandibular condyle length and homeostasis, at least in part through modulating cells that reside in the tendon to bone interface. So with that, I really want to thank all of my previous lab members and my mentor, Brendan Lee at the Baylor College of Medicine. Carolina Leynez was an amazing postbac who worked on some of the micro-CT imaging data, which I really didn't have time to get into even further, but the segmentation and all that magic was done by Carolina, and she's currently applying for medical school right now. So just FYI, I want to thank our collaborators at UCLA and U of Washington, as well as Ronan Schweitzer for helping us with the mouse lines, particularly the Scleraxis Cree and the Scleraxis GFP mouse model, and also obviously NIH for funding.